Hello and welcome to all of you who are attending this session. And as we all know, this session is about demonstration of virtual labs on DIKSHA and ePathshala AR content. AR means augmented reality content. So as we know, CIT NCRT is working towards a lot of initiatives, lot of newer tasks as per the recommendation of NEP 2020. Development of immersive content is one such need that we need to cater to, and Central Institute of Educational Technology NCERT is constantly working towards it. And as per the recommendation of NEP 2020, we are also into developing virtual labs on DIKSHA. To achieve the recommendations and goals of NEP 2020, we are also partnering with a lot of organizations and institutions. For the development of virtual labs on DIKSHA, we are partnering with Amrita University and CDAC. And we have hosted all the content which is developed in collaboration on DIKSHA. So today's session is about demonstration of these virtual labs which exist on DIKSHA, and to disseminate this information to teachers and all our viewers so that they can get benefited by the availability of these virtual labs. The purpose of having or developing virtual labs on DIKSHA is to provide anytime, anywhere access to lab experiments and activities. As we know that there is no uniformity in the infrastructure which is there for having lab experiments in the schools, and also labs have limited time access to our learners. Having all these experiments in the virtual formats ensures the availability of these content anytime, anywhere, and using your mobile also. So on these virtual labs, we have theory of each experiment, then we have animations. Apart from animations, there are simulations where we can actually handle the apparatuses and carry out relevant experiments in a virtual mode. And there is also a provision of the getting feedback and also assessment. So these are various components of virtual labs. Apart from this, there are a lot of content which are there in the textbooks which need 3D visualizations to understand the concept in a better manner. To achieve this objective and to ensure 3D visualization of abstract concepts, there are a lot of augmented reality contents which are linked to NCERT textbooks, and there is a specific app which has been developed to scan the images which are there in the textbooks and get a 3D visualization using augmented reality possibility which is there. So this is about a short intro of the demonstration session that we are going to have today. We have with us an entire team of young professionals who will be taking you through the virtual labs on DIKSHA and also ePathshala augmented reality content. So not spending much time on providing introduction, I'm handing over to the team so that you can get a feel of these simulations and augmented reality content. Over to the team. So we can start from Dr. Yash first. Over to Dr. Yash.
Thank you very much, ma'am. So my sincere namaskar to all of you, and very welcome in this training session on the ePathshala augmented reality and virtual lab on DIKSHA. So I will be demonstrating the augmented reality content, that how you can use these innovative techniques to teach to your what's the learning, teaching and learning by using this type of content. So what is augmented reality? Okay, so for example, you are going to teach any concept, and that concept is being augmented by using the 3D simulations, the 3D models, and present it to the user so that it's part of their environment. And definitely you must have seen the various apps around that show the augmented reality content. On social media we have a lot of content. The people are sharing the images with the lions and tigers in their rooms, in their classrooms. The teachers are also enjoying them. But how we can use this for the teaching learning process, and specifically we have designed the content for NCERT textbooks that is aligned with the NCERT textbooks. So one of the specification of the augmented reality is that your phone, smartphone, it can work with any smartphone. Specifically we have developed for the Android, having the version more than 4.1 and above, and you definitely need the camera to scan that image. So let me show you the app first. Okay, so we have a very simple menu of the app. You can see over there that you can choose the class. As currently we are the content is available for class 9th and 10th. Okay, and then you can choose within that class you can choose the subject, and within subject you can choose the chapters, and you can choose the topic. And when you are choosing any topic, okay, so definitely you need to download that topic from the internet, and the maximum size would be 5 or 6 MB or so. Okay, so for example I'm taking this class 10th, and definitely it's a science, and then the chapter out of, say for example, the first chapter is the chemical reaction, and then I need to choose the topic, say for example warning of magnesium rebirth. So you need to scan this image which is available at the front end. So the image, actual image from the textbook. Okay, so it means you should have the textbook with you. The textbook, if you don't have the textbook, you can also use the soft copy. Even you can use the scan copy, or you can use the printout of any of the textbook. Okay, so it will seamlessly work with all type of images, but definitely you require specifically this image. It doesn't matter in any format. ठीक है, आपको यही इमेज जो है वो टेक्स्ट बुक से स्कैन करनी है. So let's load the activity. Once you load the activity, so your camera will automatically be opened. Okay, so now you need to scan that image from the textbook. You can see I have this textbook with me of the class 10, and the chapter is 1 and the image is 1.1. So once you scan this image, your simulation will be live on your mobile phone or the tablet, whatever you are using. Okay, so we have one AR camera. If you click on this AR camera, the background will change accordingly. Now you can perform this simulation.
And laboratory coat. Click on the continue button to continue.
Okay, so we have the learning objectives. If you want to hide this description, we have a small menu at the bottom. You can click on this one, and this will hide this the typing things. Okay, and if you want to repair this one, you can again click on this one. So now you need to perform the simulation, and the simulation is very interactive actually. You can use it to any site. Okay, so
click on the sandpaper to rub it on the surface of the magnesium ribbon to remove the dirt from it.
Okay, so now you will see that sandpaper is being highlighted. It means you need to click on this one.
Magnesium burns due to its reactive nature.
You can see it from
oxygen.
any side. Okay, you can rotate this one and
click on the continue button to click on the tongs to hold the magnesium ribbon on the flame till it catches fire.
now click on the tongs.
Observation. When the magnesium ribbon starts burning, the bright white dazzling light is produced along with formation of white powder of magnesium oxide. Caution: use safety glasses to see the flame. Click on the continue button. When magnesium ribbon burns, it combines with oxygen present in the air and yields magnesium oxide in the form of white powder as shown in chemical equation. Click on the chemical change. Since in this change a new substance, magnesium oxide, is formed with different properties and composition from magnesium and oxygen. It is a chemical change. Click on Learning Outcomes. We can observe that magnesium ribbon burns with the bright light and produces white powder of magnesium oxide. We can explain that burning of magnesium ribbon is a chemical change. We can use scientific convention to write chemical equation for burning of magnesium ribbon.
So in this way, you can attempt the full 3D simulations while using your mobile phone. So let me have one more example. So for example, you are going to teach the concept of digestive system, which is available in class 10 and chapter 6. So now you need to open your textbook and open that particular image of the alimentary canal, which is 6.6. Okay, so when you scan this image,
The human alimentary canal, digestive system. Learning objective. Let us learn about the human alimentary canal. Click on any button from the side panel to learn more about it.
okay, so now you can see, so for example, you are demonstrating this human alimentary canal. So you need to scan that image by using this app, and then we have the AR camera to change the background. Just click on this one and your background will be changing. Okay, so now you can perform the simulation. Okay, so for example, you want to discuss about the alimentary canal organs, or maybe accessory organs, or maybe the digestion process. Okay, the whole digestion process you can show from this app. So click on the digestion process.
Learning objective. Let us see how digestion of food take place in the human body. Click on any button from the side panel to see the human digestive process.
So for example, I'm clicking on the ingestion.
Ingestion. The very first step involves
You can see it from any angle.
the salivary glands along to moisten and lubricate food before being pushed down into the food pipe. Salivary glands. The saliva contains an enzyme called salivary amylase. Breaks down starch, which is a complex give simple sugar. Peristaltic movement. From the mouth, the food is taken to the stomach through the food pipe or esophagus. The lining of canal has muscles that contract rhythmically in order to push the food forward. Secretion. The stomach is a large organ which expands when food enters it. The muscular walls of gastric glands. The digestion in stomach is taken care of by the gastric glands present in the wall of the stomach. These release hydrochloric acid, a protein digesting enzyme called pepsin. Pancreas. The food coming from the stomach is acidic and has to be made alkaline for the pancreatic enzymes to act. Liver. Liver secretes the bile juice. Bile is a compound that aids in the digestion of fat and eliminates digestion. The process of converting complex food particles into simpler substances in the presence of enzymes and acids secreted by different digestive organs. Absorption. This process begins in the small intestine, where most of the nutrients and minerals are absorbed. The walls of the small intestine contain glands which secrete intestinal juice. The enzymes present in it. Finally, excretion. The unabsorbed food is sent into the large intestine, where its wall absorbs more water from this material. The rest of the material is removed from the body via the anus. The exit of this waste material is regulated by the anal sphincter. Learning outcome. The human digestive system, also known as alimentary canal, is a complex series of organs and glands that processes food. The digestive system allows us to break down the food we eat to obtain energy and nourishment. It also has to excrete waste.
Okay, so in this way, you can perform the live simulation as Professor Indu Kumar has said. That's anytime, anywhere these are available. You just need your smartphone along with the internet connections, and you are connected with the 3D simulations wherever you want. So all these simulations are available in ePathshala augmented reality app, which is available on Play Store free of cost. You can download that app and try this simulation at your own places, and also you can give us the feedback. So this is all about the augmented reality resources that we have developed. Now I'm inviting my colleague Ms. Nidhi Adalakha to demonstrate the other virtual labs on DIKSHA. So now over to Nidhi. Thank you.
Good evening, everyone. So before I'll start with simulations of science, I would like to tell you that how to reach virtual labs which are available on DIKSHA website. So first of all, we will find diksha.gov.in in Google. When we get the DIKSHA portal, we have to click. You can see that there are banners of various verticals. If we click on the last banner, that banner belongs to virtual labs. So you have to click on explore icon, and when you click on explore icon, we reaches on the landing page of virtual labs. As we scroll down, we can see that information regarding virtual labs is given, that why virtual labs are required, how virtual labs would prove helpful for you, for all the students, teachers and parents who guide their children, how they can make education, how they can make learning impactful. All this information you will get in this. And when we scroll down further, you can see that we have got e-content which are available on virtual labs related to various classes. So if I click on any of the class, like for example, if I have clicked on class 11th, you can see that Here, Hindi medium and English medium appears. You can choose your medium of study. So if I am choosing English medium, then further you can see that various subjects appear. You can choose the subject which you wish to study at that time. So if I click on biology, I have landed on the table of contents of lab manual of biology. So I can search the experiment which I want to study, and after that I can click on that. So for example, if I click on for finding resources for each experiment, I have to click on this arrow. So this will show that various resources are available related to this experiment. So if I go to one of the PDF which is given, it shows another link. So now if I click on this link, it leads me to the experiment's various resources. So here I would like to introduce you what all resources you can find on virtual labs on DIKSHA. First of all, you can see that there is theory. So theory is meant for providing basic information about the concept related to that particular experiment. So you can go through theory so that you can get to know all the information so that you can understand what you are supposed to do in the experiment. Afterwards, the other icon is procedure. So in this, you can see that materials which are required for performing this experiment are drawn and are labeled as well. So you can go through this, and afterwards real lab procedure is given. Real lab procedure, that means if you are physically present in laboratory, what all steps are required to be performed, these are given here. After that, simulator procedure is also given. So this simulator procedure is given because we have one of the resource, that is simulator, available on virtual labs. So how to perform experiment on simulator, all the steps are given here. So you can read all these steps, and after that you can perform experiment using simulator. We will reach simulator. Along with that, you have other resources like you can see video. In video you can get to know the concept in the form of actually the person is performing an experiment, and you can get to know how to perform experiment in real laboratory. Then there is Viva Voice, which helps you in assessing yourself. You can answer these MCQs, and after answering these MCQs, you can click on the submit button, and you will get to know whether your answers, which answers were correct and which were incorrect, so that you can study further and improve yourself. Afterwards, certain resources are given which can help you in providing further more information related to the same concept. And then there is feedback icon also, where you can give your feedback, whether what you liked in the concept, in the experiment, and what changes if you wish to require. If you require any kind of change, then what kind of change you require, all that you can put in this. You can rate the content, and afterwards you can submit. Now let's go to simulator. So I am going to the simulator of this experiment, which is rate of respiration. So just like we respire, plants also respire, plants also release CO2, and seeds which are germinating, they also release CO2. So we need to find out rate of respiration, which we can find with the help of finding out how much carbon dioxide is released. And that quantity of carbon dioxide released is assessed by getting to notice, by noticing that what is the level of water initially and how much it rises. By finding out the rise in level of water, we can find out how much CO2 has been released by the germinating seeds, and from that we get to know whether the rate of respiration is high or low. So for doing so, the first step you can do is here in one of the corner you can find select the seeds. If you click on this, a drop down menu appears. You can click on any of the seed, you can choose any of them. For example, I have chosen groundnut. Now here we have option of how many seeds do we require. So here you can see in this conical flask right now we have 10 seeds. So if I wish, I can increase seeds. I can increase seeds to maximum level here, which is 50. So I have increased to 50 right now. Then temperature, because optimum temperature is around 20 to 30. So I'm keeping it 25. Now I can click on start. But before clicking on start, just notice what is the initial water level. So if you notice, right now it is 2 ml. So now I'll click on start, and as the rate of respiration will be recorded, do notice how much